weird little quirk from the first half when I went to shut down the recording. On this screen here, there was a weird message. And when I went to look at the video, the last part, which apparently is when I went to showing the pictures of the male reproductive tract, all that stuff didn't get captured on the screen. I didn't check the audio for that end, but so I don't know what's going on. But hopefully the second half here will work out fine. So like I said, what we want to deal with here is the female system, and I showed you in the model there, which definitely wasn't captured on the screen, the basic parts. This is a mid-sagittal section showing the main parts. They point to the ovary here, which is a little strange because it's actually not on midline, and I'll show you the next picture where it is. But they're depicting it here in this sort of light blue color. The uterine tube or fallopian tube basically connects the ovary to the uterus, which we see here. The uterus has a very thick, smooth muscle wall, so it's a very powerful muscle necessary for childbirth. It ends with this opening here called the cervix, which leads into the vagina, another muscular wall, which continues up to the outside opening. This is a anterior view of the structures. So here we see the ovaries again in that same kind of bluish color, but they are off midline. So the other picture, I don't quite know why they were in there at that point, but the uterine tube or fallopian tube we see here connecting to the uterus. The opening to the the end of it next to the ovary isn't connected to the ovary. It's actually, there's a little gap between the two. At the sort of trumpet-like ending to the uterine tube here are these finger-like extensions called fimbriae. That's plural, fimbrio would be a singular, which will move and kind of coax the egg cell into the tube after it's been released from the ovary. And then it goes to the uterus, which we see there in the middle. And then here we can see the cervix opening up into the vagina. I forgot, it skipped this picture here. This is a representation of the meiotic divisions that we see giving rise to the eggs, very similar to the one I showed you for sperm a little bit ago, with a couple of differences, or several differences. First off, the stem cell that gives rise to these is called the oogonium. In Greek, the root oo means egg. So this is the egg stem cell. It divides through mitosis into two cells that have pairs of chromosomes. This cell here that continues on is called the primary oocyte. They put the name over here, I think, just because the stoplight thing over here is on the way. So this is the primary oocyte. It's going to initiate meiosis, and then it's going to sit and do nothing for a while, and then it'll resume meiosis, where it then divides into two cells. These cells are not symmetrical divisions. One cell basically gets all of the cytoplasm, and the other cell is sort of the leftover DNA. But this is where we have just one of each chromosome here. They're still duplicated at this point, but they're separated out. The first polar body is sort of the throwaway aspect, the extra DNA that's no longer needed. And the secondary oocyte is the copy of the DNA that's kept here and all of the cytoplasm. This then goes on to divide again, giving rise to two asymmetrical cells and another polar body. And then this is the egg cell, which is called the mature ovum. Ovum is just a Latin word for egg cell, as opposed to oocyte. Now, in this, there's two lines separating out parts of the sequence. The primary oocyte begins meiosis, but stops right away in the first phase there, called prophase, and sits there for a while. It stops at that point before birth. So the oogonia divide and make all of the primary oocytes in the embryonic ovary before the girl's born. All of the cells that she's going to have that lead to all of her reproductive cycle are present before birth, and they sit there and do nothing until after puberty. Some of them are going to be used as soon as puberty starts in, but some of them might actually sit there and not do anything for decades. The exact number for what I'm about to say, I don't remember if I have it quite right, but I'm in the right neighborhood, I know. About 400,000 cells are made for this process. If a woman is in her reproductive years from the age of 15 to the age of 50, which are not necessarily the most common work years, but just a nice round number of 35. If she's producing a single egg every ovarian cycle, every 28 days, she's going to be producing about 420 to 450 cells, depending on the exact length of the cycle. She has 1,000 times more cells than that available in the ovaries. They're all present at birth, but there's plenty to go through all of her reproductive years. It's not actually just one every cycle. Several will initiate the cycle. So, but there's still enough to handle, 10 cells going through each cycle, which is not quite how many there's going to be, but maybe half a dozen or so. There's plenty of cells available for this, but they're all present before birth, and they don't continue through the rest of meiosis 1 until they get a signal after puberty, the hormones that we're going to talk about a little bit later on. The other thing in here is the secondary oocyte. When it divides, that only happens after the sperm has reached the egg cell. So that's not technically fertilization. Fertilization is when the sperm's nucleus and the ovum's nucleus come together. But the secondary oocyte isn't going to divide and make that final nucleus ready for fertilization unless the sperm is present. If the oocyte's never exposed to sperm, it never completes that second division of meiosis. So these lines are just showing a couple of points where the process kind of halts and waits for the next signal. So this picture here, I pulled off the, sorry, hit the wrong button there. But actually, I kind of want to get away from this. I pulled off the internet because it's sort of a common representation of the process of follicular growth. And actually, just this morning, I was looking up videos of this. So let me pull one up. This is not actually the one I want to play. Where was it? Came up when I looked at this. Sorry. There it is. Okay. So this is
a primordial follicle.
Okay. This is not quite 2 minutes long. It's just going to show you what's happening. In the center of this is the oocyte, and it's surrounded by some supporting cells. So this is going to show what happens through the course of what's called the ovarian cycle, the progression, the development of this structure here. So let me play this real quick.
A primordial follicle consists of an oocyte surrounded by a single layer of squamous granulosa cells. A primordial follicle becomes a primary follicle as the granulosa cells become enlarged and cuboidal. The primary follicle enlarges and the granulosa cells form more than one layer. The zona pellucida forms around the oocyte. The structure is called a secondary follicle when fluid-filled vesicles develop among the granulosa cells and a well-developed capsule, or theca, becomes apparent around the granulosa cells. The theca possesses internal and external layers. The structure is called a mature follicle when the fluid-filled vesicles form a single antrum. At full maturity, the oocyte is located in the cumulus mass. During ovulation, the oocyte is released from the follicle, along with some surrounding granulosa cells of the cumulus mass, called the corona radiata. Following ovulation, the granulosa cells divide rapidly and enlarge to form the yellow corpus luteum. When the corpus luteum degenerates, it forms the white corpus albicans.
And a lot of stuff in there. So I found that animation because this sort of picture is a little bit misleading. It looks like this is supposed to be an outline of the ovary.
A primordial follicle
hold on a second.
consists of an oocyte surrounded by a single layer of squamous granules.
Sorry, I didn't know I was going to start over. Anyway, so this is supposed to represent the ovary in outline, and it shows a primary follicle that develops into the secondary follicle and then the mature follicle, which then ruptures at ovulation, and we have the corpus luteum that eventually degenerates down to the corpus albicans, which are all the steps I just showed you there. I looked for that animation because I think it's a really good representation of something that this kind of picture misrepresents. This kind of makes it look like it's moving through the ovarian cortex, which isn't really true. It's more like what we just saw, where the follicle gets bigger and bigger, and then it ruptures and it becomes that. And that really should happen just in one location within the ovary. But this is a good representation of those different structures. I just pulled this up because it's a common way of representing it. In the book, they represent it using this series of micrographs, which is a fine way to do it because it gets away from that representation that seems to suggest that it's moving through the cortex. So here we have the primordial follicle, which in the animation I just showed you, they said the granulosa cells are squamous. I wouldn't exactly call them squamous. This isn't an epithelium. But they are kind of flat in appearance. And then they become rounder, what they called cuboidal in the previous slide. And they're called granulosa cells because they look like grains. That's what granulosa means. And they grow out and make a thicker surround as the follicle gets bigger and bigger. They pointed to the zona pellucida, which is just surrounding the oocyte, or oocyte as the voiceover just said. This is an odd representation of a secondary follicle. It makes it look like the oocyte's floating in the middle, but the animation I just showed you is a better representation of that. That's really when the granulosa sort of breaks up, as they described it. Vesicles appear in that space. Also at this stage, the outer surround of the follicle becomes rather distinct, called the theca. And then this is the mature follicle, what the book refers to as the tertiary follicle. It makes it look like it's a lot smaller than this, but it's just in a different scale. So here's the oocyte right in the middle, which is this cell here, which in fact would have even grown larger. So you can imagine the change in scale from this cell to this cell, and the mature follicle is much larger. And in the animation I just showed you, it's a good representation of that. And then this is what it looks like at ovulation. Now, all of these pictures represent micrographs of follicles at these different stages. You can't capture ovulation the same way. So this picture here shows ovulation, which you'd have to see in a living ovary under a different sort of microscopic technique. So this looks very different, but the dark stuff here represents what's left over of the follicle after the oocyte and the surrounding granulosa cells, the corona radiata, emerges from that. And then this big cloud that seems to be surrounding the whole thing is some of the contents of the antrum, which is cellular debris and fluid that's inside that space, whether it's the secondary or mature follicle space. And then here we see the corpus luteum, which is the remnants of the follicle that changes a bit and becomes an important part of what's happening here. Now, these 6 pictures are suggesting what's happening in the ovarian cycle, or actually the first one is before the cycle, and the other 5 represent the cycle. The cycle's 28 days long on average. The first 14 days are when the follicle develops. So these 3 pictures leading up to ovulation on day 14 are really half of the cycle. And then this singular picture of the corpus luteum at the end here is the other 14 days of the cycle. Okay. Just not a whole lot's happening in the ovary at this point, so things look the same. There's just one picture here. While the drawing up here of the secondary follicle is kind of misleading, this is a really good representation of it in a micrograph. So we can see these spaces opening up, which is the what the animation called the fluid-filled vesicles. I wouldn't call them vesicles. That's not really appropriate. But it's this developing antrum here. So when we see any openings like that in the granulosa, that's when we're at the secondary follicular stage. And then when it opens up all the way, that's the mature or tertiary stage. Okay. So, these figures are just depicting the hormones that are at play in this. So these 3 pictures are showing 3 different stages or 3 different time points in the process. The first phase is called the follicular phase, which is everything that comes before ovulation for the ovarian cycle. And then the middle one's ovulation. And then the last one is the luteal phase, which is everything after ovulation for the ovary. All of the parts depicted here are the same. We have the hypothalamus, the anterior pituitary, the ovaries, and the uterus. The hypothalamus releases growth gonadotropin-releasing hormone, GnRH, which causes the anterior pituitary to release the gonadotropins, FSH and LH, which then stimulate the follicles, which are in the ovaries, to start developing. As they develop, they start to release the hormone estradiol, which is one of the estrogen hormones. Estrogen is just a family of hormones. Estradiol is a specific one. Estradiol feeds back to be a negative feedback signal to the anterior pituitary and the hypothalamus, to decrease the release of those hormones, it's also going to have an effect on the inner lining of the uterus called the endometrium, which we'll talk about later. And that's what's happening in the uterine cycle. The negative feedback signal here is limiting release of LH and FSH. And once estradiol levels get high enough, they suddenly switch to activating the release of GnRH and FSH and LH. Actually, in this picture here, the arrow for LH is a little bit thicker than the arrow for FSH because there's more LH being released than FSH at this stage. But that's what causes ovulation. So LH really causes the follicle to rupture and release the secondary oocyte at that point. And estradiol is still having an effect on the endometrium. After ovulation in the luteal phase, what happens, and sorry, this is a little bit smaller. I resized this picture and I didn't get it quite right. Now what was the follicle is the corpus luteum, and it's now releasing progesterone. It's also releasing estradiol, but not nearly as much as it used to. Progesterone is then going to take over the negative feedback signal that estradiol had over here, and progesterone is going to have the major effect on the endometrium in the second half here. Okay, so I'll talk about the uterine cycle in a little while, but these are the hormones that are playing a role. Okay, GnRH, FSH, and LH are the same hormones that I pointed out in the male system a little bit ago. FSH and LH, however, their names apply to what's going on here. FSH stands for follicle stimulating hormone because they stimulate the follicles to develop. LH stands for luteinizing hormone, which refers to the fact that it causes formation of the corpus luteum. Really what it does is it causes ovulation. And after ovulation, what's left over of the follicle becomes a corpus luteum. So by causing ovulation, the luteinizing hormone leads to the corpus luteum forming. So it's a little indirect, but that's where the name comes from. So those names don't mean anything in the male system because there aren't follicles and luteinizing and all that in the male system, but it's the same hormones happening, having an effect there too. Okay. I want to talk about the uterine cycle, so I want to point out what's going on here, but before I get there, they also have a picture of the ovary here. So the outside of the ovary, it has a tissue surrounding it, which is actually very similar to what surrounds the testes. And then but the tissue in the ovary is very different. The ovarian cortex and medulla that we see here is more like an endocrine gland in appearance, unlike the seminiferous tubules that make up all of the bulk of the testes, which is more like an exocrine gland. But otherwise, besides their particular functions, they're homologous glands. They're where gametes are produced. And the darker part here, this is the ovarian cortex, which is where most of this takes place. Okay. But I really want to talk about the uterus here for a second. Now, the wall of the uterus has 3 layers to it, which is actually very similar to the 4 layers of a digestive organ or the 3 layers of a blood vessel or something like that. That kind of arrangement of tissue making up the wall of an organ is very similar from system to system we're looking at. Just the particulars of the tissues that make it up and their overall structure varies. But there's an inner layer, which is essentially the mucosa, which is called the endometrium. So it has a mucosal epithelium, which happens to be a simple columnar epithelium, and its associated connective tissue. Now that connective tissue actually has 2 different layers to it, which isn't represented here, but there's the functional layer, or the functionalis, which is just a Latinized form of that word, and the basal layer, or the basalis. You can kind of think of that as analogous to the mucosa and submucosa in the digestive system organs, but we don't quite think of it that way. Then deep to the endometrium, we have that thick, smooth muscle layer called the myometrium. And then the outside of the uterus is the parametrium. Metrium in all three of these words means uterus, so it's the inner muscle and outer surrounding tissue layers. We're really just concerned about the endometrium and mostly about the functional layer, which is what's changing during the uterine cycle. Now, before I get into talking about the uterine cycle, which is going to be from this picture here, I want to take a second to talk about hormones, because as I'm talking about the uterine cycle and the ovarian cycle here, I'm also going to be talking about the hormones involved. So let me just take a second to talk about hormones. Now, this picture might seem quite intimidating, and it's not meant to be. Whenever I'm talking about chemical structure in particular, I want to show some sort of visual representation of that. So I'm going to do something like this. Also, like I said earlier, I'm trying to use copyright-free artwork, which is what this is. This has more information than I want you to know. In fact, there's only a couple of things I want to point out in here. It just happens to be a lot going on in this picture. All I want to talk about is the fact that all of these hormones, and especially the ones I'm going to talk about, are steroid hormones, and we can tell that by their basic structure that we see repeated over and over again in all of these, which comes from the basic structure of the cholesterol molecule, which is represented up here. Now, what this figure is showing is the various enzymatic changes that give rise to all these different hormones. And if you look at all the names of these hormones, you might notice some that we've already talked about in other chapters, but I'm just going to concentrate on the ones that are relevant to today. So cholesterol gets modified by one enzyme, and then another enzyme modifies that and gives us progesterone, which is one of the hormones we want to talk about. Progesterone is a precursor to a lot of different hormones, some of which we're going to talk about today, but then others that we've seen elsewhere in this in this course. Through a few enzymatic steps, progesterone gets changed into testosterone. Testosterone can be changed by one enzyme into this molecule here, which is called dihydrotestosterone, which is just another androgen steroid hormone. So it's part of the same family as testosterone, but it's only found in men. Testosterone is actually found in women also, but it doesn't have as much an effect in women because testosterone, when it's made, and in fact it's made in the ovaries, specifically by the thecal cells that were pointed out earlier, it's immediately, or actually no, I should say that differently. It's made in the follicle, but then in the thecal cells, it's immediately changed into estradiol by this enzyme called aromatase. Now, the root of the word aromatase obviously is aroma. What it's referring to isn't smell exactly, but rather what we call in chemistry an aromatic compound. Now, there are aromatic compounds that have a strong smell to them, but not all of them necessarily do. What defines something chemically as having an aromatic structure is when there's a six-sided ring that has 3 double bonds in it, okay, which we see in this ring here in estradiol. The enzyme that converts testosterone to estradiol, really all it does is it takes this 6-sided ring and changes the bond structure in it to be aromatic, like that. In doing that, it changes the two molecules a little bit. I mean, it changes the molecule a little bit. Actually, if you look at the whole structure that's represented here and the changes that take place because of aromatase, there's a difference of 5 atoms between testosterone and estradiol. Okay? But they're fairly significant changes because of how these hormones interact with their receptors. Okay? Testosterone and estradiol are very, very similar molecules, but when testosterone binds to its receptor or when estradiol binds to its receptor, those complexes are very, very different. Now, these are steroid hormones. They're lipid soluble, which means that they get into the cell and complexed with their receptor, they mainly interact with the DNA. So testosterone, the testosterone receptor complex, or estradiol and the estrogen receptor complex, bind to different regions in the DNA and cause different changes in the cell. Okay. But there's very little difference between these two molecules here, but there's a lot of difference in their effects in the body. Okay. Now, estradiol is one of the estrogens. Here's another estrogen right here, estrone. And then there's another one that's not represented anywhere in here that I can see called estriol, which I don't think is in here. But those are all closely related molecules, and they belong to the family called estrogens. Estrogen is not a hormone, it's a family of hormones. Estradiol is the most common one. And testosterone belongs to the comparable group called androgens, which also includes DHT, the other testosterone type thing down there. Now, I'm mentioning these because I want to talk about the role of hormones in the ovarian and uterine cycles. Now, to begin with, little quirk in this picture. I don't quite know why this happened, but here we have the ovarian cycle laid out from day 0 to day 28. And here we have the uterine cycle laid out from day 0 to day 28. If I were making this picture, I would have made the 0 here line up with the 0 here, and the 28 line up with the 28, just so they're the same width. But for some reason, they're not the same width. But the idea is that they do correspond time-wise. Another little quirk, and I can certainly understand why this is was chosen. The first 7 days of what's going on in the uterine, the ovarian cycle is rather complex. So they didn't squeeze it in here because they wanted to show some more detail up here at the top. But the primordial follicles start to develop into primary follicles and then into secondary follicles. Only one of them is going to get to the stage of being a mature or tertiary follicle, which is what we see here. In the early parts of the ovarian cycle, several different follicles will be undergoing changes. Only one of them will get to the point where it's going to complete the ovarian cycle. And at 14 days, it ruptures out, which is ovulation, and then what's left over becomes the corpus luteum, and then eventually degrades down into smaller structures. It's not necessarily the corpus albicans right here on day 22, or whatever that's supposed to say. The idea is just that towards the end of the cycle, that structure gets smaller and smaller, and it becomes more and more white in color rather than yellow in color. Luteal refers to its yellow color. Albicans refers to its white color. And that's going to happen if nothing occurs with the oocyte. But if the oocyte's fertilized, it becomes a mature ovum and starts dividing, and then the zygote implants into the uterine wall, the corpus luteum is going to persist longer. It's going to be there for a couple or few months, helping to regulate hormones related to pregnancy. But the normal 28-day cycle here is what happens most cycles, if your last name's not Duggar. So, Duggar joke? Come on, people. Down here, we see what's happening with the endometrium. And the way this is depicted, the lighter colored part is the functional layer, and the darker colored part is the basal layer. The basal layer doesn't really change. It's all about changes in the functional layer. The uterine cycle, again, in time lock with the ovarian cycle, starts with menstruation. So menses is really the functional layer from the previous cycle detaching and sloughing off and being released as the menstrual flow, which takes place over the first 5 or so days. And then after that, the functional layer is going to start growing thicker and thicker. the cells are proliferating, so we call that the proliferative phase. After ovulation, it might not be fully grown, but it's going to be pretty close to its full size. And it starts behaving a little bit differently, and we refer to this as the secretory phase because the glands that we see going deep into the endometrium here are going to start releasing the mucosal secretions, which are there to help prepare the uterine lining for implantation and survival of the zygote until the communication between the maternal and embryonic systems is set up across the placenta. But if nothing happens along with the degradation of the corpus luteum, the endometrium will start to slough off and we get menstruation at the beginning of the next cycle. Okay. I did skip over just to point out here, in the ovarian cycle, the first part is called the follicular phase. Oh, I did show you that with the hormone figures I was showing you earlier. But the follicular phase is the first half, and the luteal phase is the second half, separated in the middle by ovulation. The follicular phase is mainly when the follicles are releasing estradiol, and the luteal phase is mostly when the corpus luteum is releasing progesterone. The corpus luteum is also releasing estradiol, but primarily progesterone. And it's the progesterone that causes the secretory phase of the uterine cycle. The bottom half of this figure here is showing the different hormones, how they vary over the 28-day cycle. The first graph are the pituitary hormones, the gonadotropins that are released from the pituitary and influence the ovaries. We have FSH and LH. FSH levels are a little bit higher than LH levels at the beginning, and then LH levels take off and really peak, and then they come back down and everything sort of falls off. The bottom one, we have estrogen and progesterone. Estrogen levels slowly increase during the follicular phase because that's what's being released from the follicles. Progesterone levels are practically non-existent. Then they'll peak and drop off, and estrogen will come back a little bit, and progesterone will really start to take off and be the predominant hormone in the luteal phase up there. Now, these two are actually tied together quite a bit. Estradiol acts as a negative feedback signal to limit the release of FSH and LH. As the estradiol levels grow, it's going to be a stronger and stronger negative feedback signal. And so the levels, especially of FSH, go down. But then when estrogen reaches a peak, somewhere around day 10 or 12, that level of estrogen in the blood then switches from being the negative feedback signal to an activating signal, which causes the release of FSH and LH again. Now, the negative feedback signal suppressed release of those hormones, but it didn't cause those hormones to stop being made. They were just stored in the pituitary gland until the peak of estrogen levels causes that release. And the release is much more significant for the LH than the FSH. So really, we kind of don't worry too much about the FSH levels. It's the LH levels that really peak leading into day 14, and that's what causes ovulation, the LH peak that we see right there. And after ovulation, the remnants of the follicle become the corpus luteum. And so LH, or luteinizing hormone, causes that to happen. Okay. And then when the corpus luteum becomes active, that's what's releasing all that progesterone that we see there. Now, looking at this graph actually tells us a little bit about how birth control pills play a role. Your common birth control pill is usually a mixture of progesterone and estradiol as the predominant estrogen. And a lot of the common ones these days have low estradiol levels because there's some evidence that estradiol levels can have a deleterious effect over the long term. Suggestion, I don't know what the proof says, but better safe than sorry, so estradiol levels are low, which is fine because progesterone is really the main thing we want here. As if I take birth control pills. Now, the point of taking birth control pills is basically so that your blood hormone levels look like day 21 on that chart there, okay? Which is what hormone levels should be post-ovulation. So if your body has those levels of progesterone and estradiol in them, then the ovaries are not going to or not going to develop the follicles to the point of ovulation. There won't be any ovulation. The traditional protocol for using those pills is 1 week out of the month, the pills don't have any hormones in them. And that's going to allow the progesterone and estradiol levels to drop down, like we see at the end there, going towards day 28. And it's the loss of those hormones in the blood that causes the end of the uterine cycle and the detachment and sloughing off of the endometrium, functional layer of the endometrium, for menstruation. Now, there's nothing that says that it's entirely necessary for that to happen. So traditionally, the birth control pills are, I guess you could say, designed to work on a 28-day cycle. But there are other ways of taking it. There's one brand called Season Eat, where you only take the no hormone pills once every 3 months or once every season, hence the name Season Eat, limiting the number of menstruations that are going to occur. There are also other forms of hormone delivery. There's an IUD that has hormones sort of in it. I thought there was a patch, but every time I say that, I just get a bunch of puzzled looks from most of the women in the class.
Depo-Provera.
What?
There's a Depo-Provera.
So there is a patch.
There's a pouch, and there's also the insert.
And okay.
There's inserts.
All right, I wasn't
There's
sure.
inserts there.
So it's true. I guess it's just not very common. So most people are like, why did you get a patch? Or maybe they're thinking I'm saying nicotine patch or something like that. But anyways, these are different ways of delivering hormones, which happen over a longer time. If I remember correctly, the hormone-infused IUD has either a 3 or a 5 year.
One's 5.
5.
One's 5. If it's a copper one, it's 10.
Oh, okay. So that has a different. Now the copper one isn't a hormone impregnated one. The copper has a different effect. But so, anyways, a 5-year release plan, whatever, isn't going to get to the point where the levels are going to drop down necessarily. So it's not going to mimic the menstruation part of the uterine cycle. That doesn't mean that there won't be menstrual flow, there won't be blood, because the functional layer is a very highly vascularized tissue, and so some bleeding is likely to occur, but it doesn't have to be so locked into our typical 28-day cycle that we're describing here. I'm not positive about the copper one, but somebody was asking me about that last week. I think what happens is copper acts as a, what we call that? Cofactor, I guess, that binds to something, might be a hormone, might be something else, that will either affect glandular function or some other aspect of this. I just don't know what it is. Actually, the person was asking me about it and thought that it was really strange that copper would be implanted, and wouldn't that be toxic? We have a number of metals in our body. The one you're probably most familiar with is iron in hemoglobin. But there are a number of metal ions that are found at trace levels in your body. And so in this form of IUD, the copper is not a copper wire that's just spewing off copper ions all over the place, but rather it's going to control the amount of copper that's being released. and that's going to have some effect. And I don't know what it is, but those kind of ions usually interact with proteins in some way. Zinc, I know, is found associated with a lot of DNA binding proteins. Iron, which we all know, is associated with hemoglobin and the heme molecule. I just don't know offhand what copper tends to associate with, but those kind of things are going to happen. So the copper must play some role in the signaling pathways of what's going on here and can lead to having similar effects. So that's an overview of what's happening with the ovarian and uterine cycles and how they kind of match up. I showed you this picture earlier. I just wanted to jump back here to remind you. And this is actually when I introduced the term follicular phase and luteal phase, which is the first half and the second half of the ovarian cycle, separated in the middle by ovulation. And then I also said I want to come back to this figure here. Now that we've talked about the hormones in relation to the female physiology, here we see this picture. And for whatever reason in the pictures that I'm just showing you from the female system, they don't have any anatomical representation like this of the hypothalamus and pituitary, but they're exactly the same in men versus women as far as this picture is concerned. And so the same things are being released here as we just talked about. The difference is in the testes, testosterone is released, and that's the negative feedback signal instead of estradiol. They mention inhibin here just as another feedback signal. Why they don't have that into any of the pictures that I was just showing you in the female system, I don't know, but inhibin is present in the female system also and acts as another inhibitory signal back to the pituitary there. So that kind of ties up the female reproductive system and the hormones role playing in regulating that. Are there any questions about that stuff? Since I have a little bit of time, I do want to go back to something I said at the very beginning, just to sort of complete that thought. With this presentation, when I started off, I showed you this picture and I said that it's really kind of a misrepresentation. That's not the way it would really look. This was done in a petri dish in vitro. And so the number of sperm cells there is probably greater than you would expect to see in vivo in a real situation. And the reason for that is because sperm have to travel through the female reproductive tract, which I showed you with this model here. So they have to travel through all this way. Now, this model is not created to scale at all. The sperm would not be that big relative to the size of the organs or the egg cell there. Okay? They're just making them big for the sake of being able to see them in the model. But the distance that the sperm cell has to travel is actually immense relative to what it is. I have no actual idea what it is, but imagine maybe you had to walk to Los Angeles. I don't know if that's at all the right scale, but it's a really long way. And maybe that includes the male reproductive tract too. But the issue is when it gets into the female reproductive tract, it's essentially a microbe. Just like any other microbe, the female immune system is going to try to fight it off. Now, one way to do that, which is kind of a passive aspect of the innate immune system barriers, is the vaginal wall secretes acidic compounds, just like the skin does. And so the seminal fluid contains some basic compounds that help to buffer that so that the pH is neutralized and that doesn't inhibit sperm travel through the, at least the beginning of the female tract. Then as it goes along, and especially within the uterus, it has to deal with a lot of white blood cells. Okay? White blood cells are there to get rid of microbes, and so the sperm have to make it through that. gauntlet of immune system cells. Probably, and I might be kind of making up some of these numbers, but it's along these lines. Probably about 250,000 sperm cells are present in ejaculate. So that's how many enter into the female reproductive tract. Probably the number that make it to the egg cell at the end is measured in dozens, and maybe only a dozen or so cells get there. These kind of pictures are misrepresenting because these cells, these sperm cells, don't have to deal with an immune system to get through. They're delivered directly to the egg in the petri dish. But some of the sperm cells are going to suffer phagocytosis. Others are going to interact with molecules that are secreted in the uterine mucosa, those sorts of things. Also, not all the sperm cells go to the egg cell all at once. First off, they don't necessarily know which side the egg cell is going to go on, so randomly they can go into the right or left uterine tubes. So that's going to cut the numbers down by half pretty much easily there. But also, they seem to work in waves. So there's going to be the first dozen or so sperm that move on for the first wave trying to fertilize an egg cell. They might never find an egg cell. They might just get lost as far as the rest of the sperm are concerned. And then another wave will move on a little while later, which might be a day or two later or something like that. They're going to move on and try to find an egg cell later on. So not all of the sperm cells are going to make it because of the immune system fighting them off, and probably half of them just take a left when they should have taken a right, and then they also sort of break up into waves of sperm, increasing the chances of fertilization so that their timing matches with ovulation a little bit better. When I try to remember days and that sort of thing that happen, I usually get it wrong. I think it's 5 days that sperm are viable from introduction to the female reproductive tract, but I very well might have that off by a couple of days in the other direction. Now, the sperm have to swim through the female reproductive tract, and they get energy for swimming from sugars that are present in the seminal fluid, in the ejaculate. But once they get a little ways into the female reproductive tract, the seminal fluid has diffused away most cases. There's not going to be ready access to that sort of thing. So they actually can get by because there's going to be energy-rich molecules present in the uterine mucosa, secreted from the uterine mucosa cells, which are there to help the zygote when it finally implants, but also the sperm cells can use that to power their swimming. Now, that fact actually leads to an interesting study that I read about several years ago, which addressed the prevalence of male versus female offspring. Now, simply from a chromosomal point of view, there should be a 50-50 split, because as far as chromosomes are concerned, it comes down to simply how many gametes that the father produces have X chromosomes and how many have Y chromosomes. And in the way meiosis works, it is a perfect 50-50 split. Of the 4 gametes that are produced in spermatogenesis, 2 of them will have X chromosomes and 2 of them will have Y chromosomes. But actually, the split in gender in the population isn't a perfect 50-50 split. It's usually like 51-49 or something like that. It's pretty close, but it's not perfect, which suggests that there's other things at play in gender determination. So there's a study that looked at some interesting things and came up with some very interesting suggestions of what it would be. I think they're overreaching a little bit based on the data they have, but it makes a compelling story. what they did, and I think this was done in some Scandinavian country, what they did was they just followed women, asked them what they ate, and followed them for a while to see what happened reproductively. And essentially they broke the data down into two major groups, which was women who ate breakfast versus women who didn't eat breakfast. They had more data and other ways of looking at it, but the main point is really between those two groups. And what they found was women who did not eat breakfast were slightly more likely to have male children. And women who did eat breakfast were slightly more likely to have female children. Now, it should be an even 50-50 split. So the differences here are pretty minor, maybe a 55-45% split. So there's more to it than just what they have for breakfast, but it does suggest that there's something going on there. And now I'm really simplifying the study quite a bit. There's a bit more to it. But from that, essentially, they suggested that there's at work another factor in gender determination having to do with resources available in the environment. So this is the modern environment, so people can get food pretty easily wherever they are. And we're talking about Scandinavia, rather affluent countries up there. So women are likely to have easy access to nutrition all they need to. But some people are going to make a decision, to eat breakfast or not eat breakfast or something like that. And so that was the data they looked at. What was suggested by that, a woman that eats breakfast represents a woman living in a resource-rich environment. They can get all the nutrition they need to. Women not eating breakfast would represent women living in a resource-poor environment. And what they suggested was there was a reproductive strategy at play here between what's going on in the environment, what resources are available. Now, the point here, and talking about these kind of things really gets into some very speculative ideas about biology, but the idea here is that ultimately what the reproductive system is trying to do, or really an organism is trying to do, is to pass its genetic information on to subsequent generations. The more individuals in the next generation, and the next generation, the next generation, the next generation that have your genes, the more successful you are. And so in a resource-poor environment, it's better to have male children because they're more likely to spread genetic information far and wide without much of an energy commitment to that. Now, the trade-off, however, is that you don't necessarily know if your grandchildren are really your grandchildren. Okay, so now, there's no actual voluntary aspect to this, but I'll sort of anthropomorphize it a little bit more than it's reasonable. So a woman has a child, and the success of that reproductive event can be reflected in how many grandchildren are carrying genetic information from that child. And so when resources are poor, where the woman can't, where the children can't really invest as much energy into gestating children, having a male child is better because then the genetic information can be spread quite a bit wider. It's just impossible to know if the grandchildren are actually related to you because of there's no way to make sure that your son's reproductive partner is actually reproducing with your son's sperm. It's just impossible to tell that. However, if it's a research-rich environment, then the best strategy is to have daughters because there's enough resources that there's a high probability of success of having grandchildren. She's going to have enough resources to gestate children. And you're 100% sure that they have your genetic information in them. Okay. So there's a trade-off between the energy needed to have grandchildren eventually with the surety of whether those grandchildren are genetically related to you or not. The odds are good in a resource poor environment if you have a son that can hopefully spread your genetic information far and wide. You don't necessarily know if it worked, but there's a good chance that it did. In a research-rich environment, you're guaranteed of having grandchildren with your genes, fewer of them, all things being equal, but you're definitely sure that they are genetically related to you. So that's the idea there behind that. Resource poor environments are more likely to lead to male children. Resource rich environments are more likely to lead to female children. Now, it can't be a big difference because it takes two to tango. There has to be members of the opposite sex in that generation to reproduce with. If everybody said, well, heck, there's not a lot of food around, let's have sons, then there would be no subsequent generation because you need to have women to bear the grandchildren the next round. So there's just a little bit of a difference there. Now, I think they're overreaching on the data quite a bit because if you think about it a little bit more, that suggests that in countries where resources are limited, you should see more male children. And I don't know if that's true. It's actually a very hard piece of data to get to because also there are just societal issues. There are certainly examples in various countries where people will have female children and abandon them or destroy them because they want men because of some patriarchal need of reproduction. It's actually a poor decision to do that because if you have a son, you aren't guaranteed that your grandchildren are actually your grandchildren. But there is actually, evidence of that happening in some cultures. So it's really hard to get good data of what that could be predicted from what the study suggested. But it does put forth a few ideas of other things that can go into determining gender, besides just the presence or absence of X or Y chromosomes. Now, the root of this really comes down to those X and Y chromosomes. A sperm cell is significantly differently weighted whether it's carrying an X or a Y chromosome. And so they're also suggesting that what might be at the root of this is actually the energy it takes for the sperm cell to swim. And so a Y chromosome carrying sperm cell is lighter and therefore needs less energy to swim all the way down the uterine tube. Whereas an X chromosome carrying sperm is heavier and needs more energy. It's getting its energy from the mucosal secretions in the uterus. And so in a research poor environment where the woman's not getting as much nutrition, her mucosal secretions might be nutritionally limited also. So the sperm cells that need less energy are more likely to swim all the way to the egg cell. Whereas in a research-rich environment where there's plenty of nutrition, then the heavier X chromosome carrying sperm cells could then potentially swim just as far because there's plenty of energy to power their swimming, just like the Y chromosome carrying ones. So in the paper, they're kind of, I think, overstepping a little bit with the reproductive strategy idea there, but probably just related directly to what's available to the sperm to power their swimming. That does make some sense and goes directly more at what's in the woman's diet and that sort of thing. So I just wanted to kind of finish with that, coming back to this picture at the beginning, because I really think that this picture is a bit misleading. There's not going to be that many sperm cells. It might be more like a dozen or two sperm cells that are actually at the egg when it's fertilized. So that wraps up what I wanted to cover with the reproductive system. Let me just remind you, next week we're going to have the second practical. When you come in that night on this computer, I'm going to shut down your access to Blackboard, so all of the work needs to be done by then. I will be less than understanding if you come up to me and say, but I could